but I do appreciate the opportunity to talk with you a little bit today. And the idea is I'm going to give you a fairly whirlwind update on where we are with sort of nutrition therapy, I think, in CF, and then lead into the study that we did with this special structured lipid matrix supplement. And the reason we're doing this at CF meetings is because I'm happy to say that the company Envara Health has moved forward and is going to make this product available, hopefully in the next few weeks or so. So let me move on to the next slide. Good, thank you. So as far as disclosures, which are certainly important in our world today, the original study was done with a group of my colleagues, and it was NIH funding. So that's what we refer to as the randomized controlled trial. It was also supported by the NIH through all the clinical research center because the study was very complicated and patients were all brought to CHOP and stayed inpatient and had that. We had some support from the CHOP Nutrition Center. After all of the study activities were done and all the primary publications were out, I was instrumental in helping to found a company called Medical Nutrition Innovation, which now is Envara Health. And I continue to serve as a scientific advisor for them while I'm still here at CHOP. And just to mention the other disclosures that I do consult with a number of other companies that have interest in nutrition and chronic disease and health. So next slide, please. Good. So and I was going to actually turn off my video in part because it, I think, saves bandwidth for what we're doing. So as all of us in the nutrition and CF caregiver group, we're really happy that we're at a place these days where this is now fairly well accepted by everybody on our teams, that optimizing nutritional status and growth and body composition improves patient outcomes with CF. The next slide. I'd like to go back and remind us how this journey started and where we were not in this place so much when we were in the early 80s and 90s. This is a pivotal paper, and if you've never read it, I really encourage you to do it. But this was the work of Mary Corey that was published in 1988. And I say pivotal because this really was an epidemiological comparison of the US and Canada, and then also the CF Center at Sick Kids in Toronto and Boston Children's. And this clearly showed the data that the better your nutritional status, the better your survival. And this is, one of the summary slides that shows the percent survival by age. And we were able to show, if you look at the green bar with the green shading, that's the median and the standard deviations, or the percentiles for statistical significance. And if you looked at Toronto versus Boston, or in essence, Canada versus the US, you were able to see changes with survival. And these were all healthy CF babies at birth, not those born with complications. So the next slide. And that really then put us, I think, in a very strong position with our pulmonary colleagues and infectious disease colleagues to start to really focus on the nutritional care. Now, most of the nutrition impact of CF is related to the pancreatic function issues. And so I wanted just to show this to remind us that in the pancreas, the two components that lead to our pathophysiology really relate to the special cells, the acinar cells, which are shown here. And those are the cells that really manufacture, store, and then secrete the digestive enzymes. And they go from the pancreas into the small intestines. And it's the loss of this, because these acini are destroyed and become very fibrotic, and cease to produce the digestive enzymes. For us in CF, the most important enzyme loss is that of lipase, which digests the fat. And also to mention, because it is important, there are also duct cells, and these are the cells that secrete the bicarbonate that then help regulate the acidity in the stomach, in the small intestines. And this is an issue also about digestive enzymes, particularly from the medication, the pancreatic enzyme replacement therapy, because for them to do their very best, they need to be in a specific acid-base environment. So next slide. And again, the fundamentals that it's very well established now that the vast majority of people with CF have pancreatic insufficiency. And it varies a little bit by geography, but in the US, it's really been around 90% for a while. And depending on different genetic groups, it can be a little bit higher and lower. But our story really is primarily from a nutrition point of view, pancreatic insufficiency. And it's extremely common in our patient care group. Next slide. So then again, to frame this a little bit, what happens when you have a lifelong persistent malabsorption of fat? So that's usually, we are concerned mostly about calories, but fat soluble vitamins as well. So in this slide, we're looking at the birth cohort from the CF, the patient registry data set, which again is an extraordinary resource. Going back to 1989. And on the top of the slide, you see the trends for weight. And I've highlighted the 50th percentile because our goal really is that our patients with CF would have normal weight and that the trends ultimately are to be at or a little above the 50th percentile. And then we would like to bring those down in the low percentiles up over time. Then looking at the height trends in the same format, you can tell we've actually been making good progress with weight gain in certainly some of our patients. But when you look at the height trends, it's clear that we still are not getting the patients enough nutrition support for them to grow in stature to their genetic potential. And just to mention here that there's nothing in CF that really is coding for or genetics for short stature. And in fact, if anything, because a lot of the gene pool comes from people with more Northern European heritage, the actual, when you look at the genetic potential based on the biological parent heights, that it's even more profound that we're not hitting that benchmark of normal height growth. So next slide, please. So this I wanted to, because we're all so interested and really fortunate now with newborn screening, we actually get to meet our patients at a very young age now. Often now with newborn screening, they're identified and in clinic by 4 to 8 weeks. And we do anticipate that's going to make things better and better. But here looking at, in essence, diagnosis up to 24-month cohort over the last 20 years, you see the same thing that I was talking about. What I want us to always do is, look at the weight and height percentiles first, because that's really the growth of the individual patient. And even though I'm certainly one of the proponents of using BMI or BMI percentiles as the screening tool and as one bit of data, we always have to remember that the BMI is a derived variable. And we have to go back and say, well, how is the height and how is the weight for the individual patient? And again, in classic nutrition and malnutrition, we know that you have to have a good weight status persistently for you to be able to kick into all of the physiology that lets you grow at your genetic potential. So it's expected that the height improvements are lagging the weight improvements. Okay, next slide, please. Thank you. And then just to end this, section, because like I said, one of the things that happened that really was another game changer for us was to get the data and the evidence that showed, if you look over on the left, looking at the percentile for BMI in children and young adults, that we did see changes in their ability with their weight and what happened with their FEV1 and their lung function. And so that became part of our standard of care now, where our goal is to get children's BMI at or above the 50th percentile. And again, I'll call your attention to, even though the BMI is over the 50th right now in this data set, it's because the patients are short. And part of our goal is to get them to grow to their genetic potential. And now certainly that half of our patients are adults with CF. We also really were able to focus on BMI goals or weight for height goals for adults here, the BMI of 23 for males and 22 for females. Next slide. So now to really go and focus on the study I wanted to tell you about and how that's led to where we are, I think, with having a real innovation that's going to help us treat people who are underweight and children who are under height with CF. So the background of the study really is what you all know, that even regardless of using enzyme replacement therapy in the ways we know how to optimize it, that there is still fat malabsorption in most patients with CF and pancreatic insufficiency. And so this product that I used in the clinical trial, which is based on a structured lipid matrix, in the studies you'll hear it referred to as LXS or Limixorb. Now that's what you will learn about later as Encala. But the inventor was really able to come up with a very specific approach to really optimizing absorption of dietary fat, really without the need of lipase. And it's based on a combination of lysophosphatidylcholine and some specific fatty acids, including the two essential fatty acids. So this is what was tested in the clinical trial. Next slide, please. So the original trial had 110 patients enrolled, and it's really the largest, or one of the largest nutrition-focused studies that's ever been done. The patients that came and volunteered to be subjects came from about 10 different CF centers up and down the East Coast. They all came to Philadelphia to participate in the study and came 3 or 4 or 5 times over the study. The major finding for what we're going to be talking about was randomization and use of what's now Encala was shown to improve the coefficient of fat absorption from a mean of 82 up to 88 over 12 months, along with some other positive outcomes. We certainly don't use CFA in clinical practice, and I'm sure most of you know that's where you collect all of the dietary. Whoops, I'm getting a little, okay. You collect all of the stool over 3 days, and you collect records of all of the food and beverage intake that was related to that stool. And this is the gold standard test, and in fact, it's what the FDA used and continues to use when it's testing, for example, a new pancreatic enzyme product to see if it's efficacious. So even though this isn't a clinical outcome, it is the gold standard, and we were able to use it sort of in this very specific research setting. So what I wanted to spend the core time now is really about a secondary analysis that we did just in the last year, because the question that came up that I couldn't answer from what we had already published was, if do patients with more severe malabsorption have more benefit from Encala. And we were then able to go back and design a secondary analysis that is based on the subset of subjects that had really sort of perfect data for the CFA and over from baseline to 3 months, and had the other important outcome variables that we wanted to evaluate. Next slide. Good. So this is a slide from the original study with 110 subjects. And what this was sort of the end of that phase of analysis. And this was part of why I realized that we actually had hints that the people who had the most improvement were going to be showing really clinically important outcomes. So this data set is set up to show you on the left total fatty acids, so all of the measured fatty acids from plasma. And then on the right, looking specific at linoleic acid, the more important or the more predominant essential fatty acid. And I asked the question is, if you had a big change in your total fatty acids or in linoleic acid, how is that related to changes in clinical outcomes of interest to all of us? And you can see here circled in red the ones that were statistically significant, across these two outcomes. And, weight for age, actually height for age did not come out significant at this point, but BMI did. And fat-free mass, which we're all getting more and more interested in these days as we move from thinking, I think with more precision, not just about body weight, but how much of that body weight is going to be muscle and fat-free mass, and how much is going to be important fat stores and where those fat stores will be. The next slide just carries on this, because what I then was looking at as well, are there trends that didn't hit significance, but that we might think about? And here you see now we populate, again, almost everything of our interest is showing some positive trends towards, if we could make total fatty acids in the blood better, is that likely to be associated with, for example, FEV1 and pulmonary function. So this, again, was sort of the close of the first study. And then the next slide, just then to give you the details of this last paper that was published this year, I think in probably May or June. So here, what we did is look for the patients who had a complete baseline visit data, including that very important outcome CFA, and really very complete 3-month data. And what we had was 66 patients then, again, all as with the original study, all very much guaranteed to have pancreatic insufficiency. And our age group at enrollment was 5 to 17. And, and this is the reason I underline the active placebo point here is it's important to tell you that this was a study comparing the new structured lipid with a very active placebo, standard of care, and not with a low-calorie placebo. And we designed it so that the structured lipid and the placebo had the same number of calories per dose and the same amount of fat per dose. So that allows you to try to isolate the findings that really are more attributable to Encala, not just to getting more calories. And then we looked as outcomes as weight, height, and BMI, Z-scores, and again, the plasma fatty acid profiles and the CFA. So to do this, as I said, we had 66 patients, and to do this kind of work, we chose the median CFA of this cohort, and then categorized those that had lower than the median, which turned out to be 88%, as the low CFA group, and then higher. And the low CF group, of course, is those who are at higher risk for malnutrition related to fat malabsorption. Okay, next slide. And then just to show you a couple of data slides here. Again, on the left, you see the N of 110. And I wanted to point out, again, this is the baseline in the 3-month data. So when you took the whole group, it's always the good news is at baseline, the placebo and Encala groups were not different at baseline. So randomization worked. And then looking at the 3-month data, you can see that the active placebo group stayed about the same, 84 to 85% CFA, and the Encala group improved from 82 up to 88% fat absorption, and that was statistically significant. Now, when we took that same approach and looked at the patients in the secondary analysis, so now the N of 66, and we focused on those with the greater need, those with the lower CFA, the higher malabsorption. Again, the good news is that baseline, even though there was a little bit more numeric difference, this was not statistically significant. The 72, 73 versus the 78, 79. And then after 3 months, you're able to see that the placebo group basically did not change in their CFA, the 82.5 to 71.1. However, the Encala group really did improve, and this is up to the 86.3%, and now highly statistically significant with 0.002. So this really did show the impact, being able to isolate the impact of just having more calories to the more effective calorie absorption, fat calorie absorption from Encala. And just to remind you that during this study, all of the patients were on PERT. All of their CF care was really managed at the home center. We did not change, part of the study, we didn't change any other directions. So they really were on usual care. They had access to their usual team, and that was part of the baseline. But with this, we were able to show those with greater CFA improvement really were those on Encala. Next slide. Okay, so now looking at just the group of 66, again, the highest risk. If you look over on the left side, this is the data set from the 2 essential fatty acids. So you see the linoleic acid and the alpha linoleic acid as our 2 essential fatty acids. And placebo is now in the gold color and Encala in the blue. So here, instead of baseline to 3 months, this is showing the percent improvement in that highest need group. So in linoleic acid, the placebo group just changed 4%. However, the Encala group improved the plasma concentration of linoleic acid by 51%. And in alpha linolenic acid, you see the same pattern. And both of these changes in Encala were highly significant. And then if you go over to the panel on the right, here's now looking at the growth outcome data over this 3-month secondary analysis. So the way I set it up is looking at the Z-score at baseline, and then what was the Z-score after 3 months, and then what was the change in Z-score. Because, you know, minus Z-scores are suboptimal. And so as these minus 51 BMI became minus 31, that was a change of 0.20, and that's moving in the right direction. So I'd like for us to, again, because I really want us, particularly for the children, we're really thinking about the growth outcomes as well. So if you look at the bottom line, which is the height variable, you see that there was a change in a positive change, an improvement of 0.06 z-scores, and highly statistically significant. With weight, even a bigger change in the z-score of 0.17. And then, of course, the BMI, because both of those have improved, the BMI has gotten better as well. So all of our anthropometric elements are moving in the right direction. So when I look at this, I mean, again, we come back to there is, the difference between placebo and Encala, I think, shown really quite well. But the other thing that was exciting, I think, clinically and looking forward, is that we got improvements in both weight and height velocities. So we really are seeing catch-up growth in this group of patients at highest need. So next slide. Now I wanted to, again, I think in this kind of a setting, I don't want to go through all of the data slides, but to really tell you everything is there in the publication. But I thought it was interesting when, so this is the two CFA groups in the secondary analysis, the less than 88 and the greater than 88. So it's interesting. I mean, it was sort of as expected, I think. The low CFA group are older, and we know clinically and child development wise, as kids get older, they're harder to get to do everything you want them to do for CF care. But we did see that, and that was clear. And that, again, at baseline, you really can see the real difference between the BMI in these two groups. If you look at the BMI z-score, those with good fat absorption have a BMI of close to 0, which is for a group of people exactly what you would want, whereas the more at-risk group had a z-score of about minus 0.4, which is, for a group, again, a real difference. The stool fat, this just brought forward when we did the analysis on the stool, sort of from a quantitative way, what was their loss? Well, if you're known to have good fat malabsorption, you're not malabsorbing much, like closer to the 8 grams per day, whereas the ones in the at-risk group were up at 24 grams per day. The CFA difference is, of course, completely driven by the study design, where we divided people up. But I thought it was interesting to see that, in some patients, we are getting really good fat absorption. Whether you use the 92% as sort of healthy person or 95, 96%, which has sort of been proposed as well, that some of our patients are doing well and growing better, and then how low it is in the at-risk group. And then the last data point, because we're all going to be the nutrition folks, the question I often have is, well, what are people really eating? And again, the diet records and from this study really provided us some very good high quality data. And you see that the people who are growing better and have better fat absorption patterns actually eat more. And completely anecdotally, but I attribute that a little bit to that if you're feeling better and you're not having as many stomach aches and your fat's being absorbed well, and of course you've had access to great CF nutritionists who've been teaching you all along about how to optimize your diet, that we can get people up into the eat more food if you have to. And that it's certainly lower in food intake in the low CFA group. And remember, these are a little bit older kids as well. So next slide. And this was, again, just to mention that in the paper you'll have the full data set comparing the groups on all of the growth data here. And then the next slide. This is the detailed data now looking at the plasma fatty acids. So here, what I just brought forward to show you is the two essential fatty acids, and looking again at the differences that I showed you on the bar charts as well. But then we've also been very interested in making sure that we're improving the polyunsaturated and the monounsaturated as well. And then the total fatty acids in the plasma, again, is an indicator of what was absorbed, in the last meal or two when we were doing these kinds of studies. Good. Next slide, please. Good. So I wanted to add just a couple of other parts about the new product and mention that before we go to questions and discussions. This is what the packaging is going to look like. It'll have the nutrition facts, panel as it should. The exciting thing, one of the exciting things for me is, since the product I had for the clinical trial a few years ago has been improved in a number of ways, but one of the really good ways was to get the wheat out of it, because part of the coating we used in the trial had some sugar and wheat to help keep the fat into sort of a powder form. So now we're at a place where this is a gluten-free product, and also we've optimized the fat content, and you can see that the 100 calories really is mostly coming from the 6 grams of fat, and the total carbohydrate is coming from a tapioca kind of powder coating, again, to keep it distributed, and is really not considered added sugar. So the product that will come to market soon, I think, is better designed than what I had as the clinical trial prototype. Next slide. So a little bit just to review how we got here with this. As I mentioned, the product, the idea and the product was invented by a Dr. Yerser, who was very personally committed to CF and CF nutrition through, as often happens, family, friends who were affected by the disease. And it was based on this lipid matrix, and I included the little cartoon kind of molecular structure before, and that this is really different than anything else that we've had for therapeutic nutrition. Often the term structured lipid is used just in relationship to change the fatty acid on a triglyceride moiety. But again, this is really quite different. It's the lysophosphatidyl based story. And now, so we've shown that it improves dietary fat absorption, that it is really lipase and bile acid independent, so that not that you still need enzymes for the rest of your food, but for this highly efficient absorption of this specific fat, you would not. That it improves fat soluble vitamin absorption. It's a very choline rich product, and again, I think someday in the future we're going to be thinking more and more about choline health in CF, and that metabolic aberration has been noted for a long time. So the choline will directly address that as well. It is a medical food, and it's a medical food that was, if you will, manufactured using all grass and feed ingredients, so generally recognized as safe FDA type classification. And then as we originally with the study, and I think again better now than we did, The main thing is that it would be easily absorbed. It's healthy fats. It's calorie rich. I mean, that's the therapeutic drive. The other good things is that it's a very taste neutral powder. So it was designed for longer term use. And the way we did it in the trial, and I think the way it will go forward, is that the patient decides which foods and beverages they want to put it in. And there's a big variety of what can be used. And the doses, whether you would be recommending and prescribing 2 doses or 3 doses, possibly 4 doses a day, really doesn't have to be monotonous. And in our experience with our 100+ kids, they came up with a lot of creative ways of doing this. Just to mention the product, because of the source materials, it's really all plant-based. And as I mentioned, it's gluten-free. And now all of the, if you will, the raw ingredients are going to be non-GMO. So I think some of the things that we and our patients would want to have sort of sorted out before it came to market, I really do credit EnviroHealth with getting that done.